This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 364, recorded on November 20th, 2015. Hello, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How you doing there, Kathy?
Doing great.
Thanks for doing this. And it's an unusual hour again, 10:30 in the morning, so our feng shui is all messed up, but we'll deal with it. Also joining me from North Central Florida, Rich Condit.
Hi. Not really. I'm in Austin, Texas.
Yeah, that's right. From Austin, Texas.
My home is in North Central Florida, but I'm in Austin, Texas. And I'm even an hour earlier than you. But my feng shui is just dandy.
I think yours is always dandy.
Let's roll.
All right. Today we have two special guests to discuss a paper published very recently. Online was published 9th of November. This paper has generated a lot of press, in my opinion, much too negative, but we'll let the authors tell us about that. We have the last and first authors, and the name of the paper is A SARS-like cluster of circulating bat coronaviruses shows potential for human emergence. Joining us, he's a professor at the University of North Carolina, Chapel Hill, Ralph Barrick. Welcome, Ralph.
Hello. Hey, Vince.
Thanks for joining us. And the first author on the paper is a postdoctoral fellow in Ralph's lab, Vineet Menachery. Welcome, Vineet. Did I pronounce it terribly?
I've heard much worse.
Menachery or Menachery? Is it Menachery, right?
Menachery.
Thank you. I completely understand mispronounced names, as you might imagine. This episode of TWiV is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai, composed of over 20 virology labs, all in one building in the heart of New York City. This department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to the program. The deadline is December 1st. Not too far off, but you do have some time. If you're looking for a postdoc, you're encouraged to check out their website and contact your faculty of interest directly. For more information about the department, visit www.mssm.edu/MIC. Once again, mssm.edu/MIC. Department of Microbiology at Mount Sinai here in New York City is where I got my PhD. Many, many years ago, I graduated in 1980, 35 years ago. I loved it. I worked for Peter Palese. I was his first PhD student. And since then, the department has gotten better. Lots of great virology as well as immunology there. Great place in New York City and a big booming medical center. So if you want to do virology research, check it out. We appreciate their support of TWiV. I know Ralph has to leave in about 30 minutes, and Vineet will stay with us for a while. So let's dive into this. And maybe, Ralph, you could start by telling us what were a little background for this paper and what were the goals of the experiments.
So one of the fundamental problems in emerging virus research is that viruses exist within animal species as a heterogeneous population of related viruses. In the case of the SARS-like viruses, these can vary up to 50% from the sequence of the SARS epidemic strain that emerged in 2003, caused 8,000 infections and about 800 deaths, and disappeared from human infections around 2004, 2005. But the problem with emerging viruses is that they can reemerge, and they do reemerge periodically over time from this animal reservoir. And when they do, they're not usually the same. The sequences are different. so that vaccines and drugs that are developed against the original epidemic strain may not be effective at all. So if you're interested in developing strategies to protect the public health against future emerging strains, you have to have an idea of the high-risk potential, the strains that have high-risk potential that are circulating in the animal reservoirs. And so the goal of this paper was to try to identify strains that were existing in nature, that had the capacity to replicate in humans, and that were variable from the 2003 epidemic strain. And if we could resurrect those strains and identify them, we would be much better positioned to develop and design and test vaccines and antivirals so that they would have greater breadth and protect against future outbreaks.
And my understanding is that you were interested in the coronaviruses in bats that were SARS-like, right?
That's correct.
And you have, for these, a lot of sequences, but you don't have virus isolates. Is that also correct?
So over the past decade, there have been tremendous advances in metagenomics. And so many groups have been sequencing, in essence, bat guano or various bat parts, looking for viruses that are harbored within bat populations. And there's about 1,300 different bat species. And the current estimates based on that metagenomic analysis is that each bat species has somewhere between 5 and 8 novel bat coronaviruses. Some subset of those are group 2B coronaviruses that are related to SARS. And so over that past decade, sequences have emerged that range from 97% identical to SARS to 50% identical. And that's specifically within the spike glycoprotein.
And the focus on...
Yes, go ahead.
Pardon me. The focus on bats is because one way or another, depending on what story you subscribe to, but one way or another, SARS was supposed to have emerged from bats, right?
That's correct. So the original hypothesis was that SARS was a bat virus that was spread into civets and raccoon dogs in the open markets of Southeast Asia, adapted to those species in the open markets which were being used for foodstuffs, and then transferred the virus adapted to humans. We actually think, and several others have reported, an alternative hypothesis is that SARS was originally a bat virus that jumped immediately into humans and then adapted to civets in the open markets where a transmission cycle was set up. Obviously, in the first model, it's a two-step event requiring mutations to adapt first to the civet and then mutations to adapt to the human. So it seems like future events would be unlikely. The alternative hypothesis was that viruses were harbored within bats that had pre-potential or were pre-poised to replicate efficiently in humans and perhaps civets. And so they could move directly from bats to humans, and then interacting with the civets in the open market, set up a transmission cycle to maintain the virus in human populations. We actually, my own personal hypothesis is that the second model is more correct, that the bats actually harbor some subset of viruses that are generalists that can use ACE2 molecules. ACE2 is angiotensin 1 converting enzyme 2, which is the receptor for SARS docking and entry into cells. And we think there are bat strains that can use the bat ACE2 molecule, the human ACE2 molecule, and the civet ACE2 molecule with high efficiency. And hence those viruses would be pre-positioned to, or high risk in terms of their ability to initiate a new epidemic.
Now, in your paper, you used a bat virus called SHC014. Why did you pick that one? A sequence, I should say, because you didn't actually have the virus, right?
We didn't have the virus. In fact, no one has ever cultured the virus. It only existed in silico, in essence, as a full-length sequence. So when you model and Vineet did a lot of this work in a collaboration with some other postdocs, when you model the receptor binding domain and you look at the residues that are present that interface with the ACE2 molecule of bats, civets, or humans, there is some conservation. But there were 7 of those residues were different, including, I think, 5 or 6 that occurred at really important sites for interaction. We knew from some studies in our lab that at least one of those sites were tolerated in terms of being able, one of the mutations in that civet, in that strain, SHC014, could occur within the context of a wild-type virus and still program efficient entry into human cells. So that was interesting to us. We were also interested in the strain because it was about 12% different in the spike glycoprotein sequence. And so it may function as a bookend. In other words, if you're trying to identify what cluster of strains were high risk that could use the human ACE2 receptor, this might be at the boundary of that cluster. Mm-hmm. And the reason that's important is if you're developing vaccines or drugs, or immunotherapeutics against SARS-like viruses, you really want to capture the maximal amount of diversity that still would program efficient entry into human cells, mm-hmm. because those would be the most high risk. So it had a fair amount of variation. There was some conservation in the receptor binding interface sites. There was variation within some of those sites. At least one of those we knew probably could function and still mediate efficient interaction with the human ACE2 molecule. And so it was worth a try. Mm-hmm. There were other things we could do with that spike as well, even if it didn't program infection. We could use it to look at cross-neutralization. If we made antisera against that SHC014 spike, could it neutralize SARS? That would be another thing we could do if the spike was not capable of programming infection, capable of programming infection in mammalian cells. We could use it for antigenic cartography, those types of things. So there was a lot of things we could do with that spike. Go ahead, Rich. Sorry.
I want to make sure I have my context right for the spike glycoprotein. So this is the thing that sits on the outside of the virus and gives it its crown appearance, hence coronavirus, right? And it's the thing that attaches the virus to the receptor on the cells. Is it solely responsible for entry? Does it do fusion as well, or are there other proteins involved?
The spike glycoprotein and its interaction with cellular components is a major barrier for cross-species transmission of coronaviruses, and it occurs at least 2 levels. The first is the ability of that spike to program its ability to bind to receptors on the surface of cells of from different species. So in the case of ACE2, for example, the SARS epidemic strain can bind to the bat ACE2 receptor, the civet ACE2 receptor, and the human ACE2 receptor. It binds poorly, for example, to the rat ACE2 receptor. The second major, and so that first interaction event is key for virus binding to the cell, and that begins to program the downstream steps that are critical for virus entry into that cell. The second major barrier for coronavirus entry and cross-species transmission is proteolytic cleavage of that spike. And so the coronavirus spike is generally broken into 2 pieces, an S1 N-terminal domain of about 90 kilodaltons and a C-terminal 90 kilodalton domain. Proteases, and there's 4 or 5 depending on who's doing the study, different proteases have to cleave that spike to cause conformational rearrangements to expose a fusionogenic domain, and that fusionogenic domain that then fuses the envelope of the virus to the surface of the cell, or the surface of an intracellular vesicle to mediate entry into that cell. In the case of coronaviruses, there are proteases that can mediate that event at the surface of the cell and intracellularly. And so depending on the spike, the cleavage events may occur on the surface to mediate fusion to the plasma membrane or fusion to internal vesicles internally with the intracellular proteases. Go ahead, Rich.
So those are host proteases, and this proteolysis happens in the process of entry, not
That's correct.
before, like flu. Okay, great. I got it.
So,
Thanks.
for example, in collaboration with a researcher at University of Minnesota named Fang Li, Fang Li has shown that HKU4, which is a relative to MERS coronavirus, that HKU4 strain can bind to the same human receptor that MERS does, which is dipeptidyl peptidase 4, and it binds very well to the human DPP4 molecule. However, the barrier for entry is not in receptor binding. The human proteases can't cleave the S2 spike and can't activate the fusion domain. So the major barrier for HKU4, and the reason it's not infecting human cells, is because of proteases. Host proteases not being able to process the spike appropriately.
So I think in the interest of time, we should continue talking with you, Ralph, and then we'll have Vineet go back and go through the experiments in some detail. But your strategy was to take a human SARS coronavirus strain and substitute its spike with that of SHCO14, and you found that this could infect human cells and mice. Now, that's part of what's generated a lot of controversy about this paper, because you made a new virus, and Simon Wayne Hobson said this virus doesn't exist in nature, and who knows what would happen if it got out. And so some people have said, why didn't you simply make VSV pseudotyped with the spike glycoprotein of this virus?
So pseudotyping is a common strategy that's used in viruses to ask particular questions about the functions of surface glycoproteins. And we did experiments with pseudotyping. So in collaboration with our collaborators at Wuhan University, we pseudotyped lentiviruses with the SHCO14 spike and some other spikes from other bat coronaviruses and human coronaviruses. The other spikes could program entry into human cells, and the SHCO14 spike could not. So that data actually suggested that SHCO14 would not mediate efficient infection into human cells. Now, there are a variety of problems with pseudotyping experiments. The first is that you don't actually know whether the spike distribution on the surface is appropriate. You don't know if the number of spikes on these are the surface glycoprotein, whether the number of those spikes on the surface of that pseudotyped particle are in the right organization. And you don't know if they are functional in that context. And so a positive gives you some level of assurance that a spike can program entry into the cell. A negative doesn't necessarily mean that the spike is not capable of mediating infection. So we felt that it would be prudent on our part to more rigorously test that, and so we made the Chimera.
So Simon Wayne Hobson has said that if this virus got out, you couldn't predict what would happen in people. How do you address that?
So number one, it's unlikely that the SHCO14 spike, which as far as we can tell has never circulated through human populations, would be capable of programming as serious an infection as the SARS epidemic strain was capable of doing. We have no evidence, no idea, no understanding of coronavirus transmission or the genetic factors that regulate coronavirus transmission. Since SHCO14 had never been transmitted through human populations, it would be unlikely that it would be as efficiently programmed for causing transmission between humans. SARS in all of its glory, both in 2003 and in 2004 and 5, was controlled by public health intervention strategies through quarantine, contact tracing, was able to stop the epidemic, to stop the reemergence of the virus in 2004. Third, I think what many people don't understand is that coronaviruses undergo recombination at fairly high frequencies. So in any kind of mixed infection, 30% of the progeny can be recombinants. Mm-hmm. And in the case of many of the bat viruses in the metagenomic sequence databases, there is lots of evidence that recombination has affected the population genetic structure of the coronavirus family. And in fact, modular evolution occurs, where individual genes can be swapped from one strain to the next. Not only the full-length gene, but also functional component parts of the coronavirus spike can be transferred from one strain to the next. So even the receptor binding domains, to some extent, may be interchangeable among closely related strains. And since some bats, not all bats obviously, but some bats live in these large panmictic populations with several species cohabiting, with them harboring up to 5 to 8 different bat coronaviruses at a time. Recombination is fluid in coronaviruses, and the spike is well adapted to handle recombinant molecules. So anyone's argument that these kind of events can't occur is basically not knowledgeable about coronavirus. Mm. So this isn't something that would not ever occur in nature. It probably occurs in nature and can occur quite frequently.
Yeah. What kind of containment do you use to do these experiments?
So we use BSL-3. We work under BSL-3 settings. This is a high containment research lab under negative pressure. All the researchers in the lab wear PAPRs and TWiV suits. These are personal air breathing apparatuses that scrub the air of virus particles, so they are in essence in containment suits. All research is done in a safety cabinet. It's called a BSL-2 safety cabinet. The virus is not free outside of that cabinet at any moment. And then we have fairly vigorous reporting standards in terms of infection control, in that anyone who does come down with a cold immediately reports to occupational health and safety, for UNC. A group of physicians as well as EHS officers become engaged. We track the infection in these individuals, and most of the time this is like common cold. In fact, every single time it's been a common cold. Mm. And then in some instances, they may be quarantined at home for 2 or 3 days. In other instances, if they don't break fever, they'll just be watched. Mm-hmm. And if a serious infection occurs, they can be hospitalized. So,
sorry.
Yes, go ahead.
So these experiments began before the gain of function moratorium, if I understand it correctly, and you've had some communication with NIH during the course of the experiments since. Can you describe that?
So we had submitted paperwork to do these experiments, I believe, in April of 2014, and started the experiments in May of 2014. We had actually contacted our program officer under which this grant, which we were doing these experiments in the summer of 2014 to talk about them in more detail. When the moratorium hit, we immediately called them to talk to them about what our responsibilities would be under this pause. And I had several grants at the time, and so that different things happened. So, for example, we were one of the groups that were asked to develop mouse models to MERS coronavirus, which required passage in the mouse, and those experiments were immediately halted until appropriate paperwork could be submitted, and then we were released to continue those experiments right before Christmas of 2015. We had other grants that were funded that they would not release the funds until the paperwork associated with experiments that NIH had identified as potential gain of function were addressed to a committee's satisfaction. The committee that actually makes these decisions, I am not privy to its organization or how they deliberate. So it's a black box to the researcher. And they will either decide that experiments do not represent a gain of function. They will, or they will decide that this could be a gain of function, and they will ask for a risk assessment in terms of what we would do. So, for example, if the SHC-014 virus had replicated more efficiently than wild-type SARS virus in human airway epithelial cells, in terms of about a log titer, or was more pathogenic in mice, the way we were given approval to do these experiments, we would immediately, if either of those events happened, we would immediately notify our Environmental Health and Science Group department and the IBC. We would then immediately notify NIH. We would stop all experiments, and a group would be convened to make a decision about how to proceed. That decision would be obeyed to the letter of the law. Mm-hmm. So if that group had decided that these experiments were too dangerous to proceed, then, and they wanted us to destroy it, then we would destroy it, and that would have been the end of the experiment. Fortunately, in this case, SHC-014 replicated, the full-length virus replicated less efficiently in human airway epithelial cells, and even the chimera was attenuated in mice, and it didn't replicate any better than wild-type SARS. So that data suggested that we really hadn't gained function.
Mm-hmm. In your discussion, you write that the data are at a crossroads of gain-of-function research concerns. Maybe in the last few minutes that you're here with us, maybe you could expand on that a bit.
So at this point, the final rules that determine gain-of-function have not been established and are being deliberated by a group, I believe the NSABB, but I'm actually less sure exactly what the group is, the makeup of the group that's making that decision.
I am too. I have no idea.
So anyway, that group is going to come down with a series of rules and regulations that people are going to have to follow, the boundaries of which I don't believe have been set. So it's quite possible that these experiments would never be allowed at all. Mm-hmm. And I think that would be tragic because the only way that so we have a resource problem with emerging pathogens, and this is the same resource problem with SARS and with MERS and with Ebola or Nipah or Hendra virus. These viruses are highly heterogeneous. They exist in nature. They spill out at periodic intervals, and they cause significant morbidity and mortality and economic hardship. And a single vaccine is not going to solve the problem. A single human monoclonal antibody therapy is not going to solve the problem. We have to have the reagents in hand that allow us to develop broad-based therapeutics and vaccines and drugs. And we can develop drugs, but if we don't have highly variant viruses to test them against, we'll never really know whether they work until an outbreak occurs. And so that leaves, I think, the research community in kind of a catch-22. We have two choices. We can we can take a reactionary response or a reactionary approach where we wait for outbreaks to occur, families lose their loved ones, and the research community then initiates massive and parallel efforts to develop a drug and a vaccine that would work against this new unknown. That's a very painful setting for humanity, and that's the standard that has been used for responding to new disease outbreaks for most of our existence on this planet. The other approach is to take a proactive response, where we try to identify representative strains within this family or this swarm of related viruses. And as we develop drugs and vaccines, we now have reagents to ask whether they have breadth of activity. And so we actually have some very interesting data with new drugs that may work against SARS and MERS. And it looks like we'll be testing soon whether they would work against these related group 2B SARS-like viruses. And if they turn out to be effective, then we actually will have reagents on a shelf that could be available to respond in future outbreaks. If they don't work, we have reagents now available so that second-generation compounds could be modified and targeted so that they'd be more entire cluster. And so we're interested in that as a conceptual approach to provide an alternative future in the case of an outbreak of highly pathogenic respiratory coronavirus.
It seems to me that as science progresses, we have to change our approach, as you say. We don't use the old approach. We get proactive. And your paper, and we'll hear the details from Vineet in a moment, it so beautifully shows that there are spikes out there in bats, spike genes that are not neutralized by monoclonals. They're not taken care of by vaccines, SARS vaccines. And we need to know what's out there and as you said, make broadly active inhibitors. It seems to me that anyone who says this work doesn't tell us anything, simply hasn't read the paper or doesn't understand it.
I agree completely. And I think it's the other thing that individuals need to think about is that in the 21st century, we've had two major new emerging highly pathogenic respiratory coronaviruses appear in human populations. And SARS coronavirus is one, and MERS coronavirus is currently the second one, and that outbreak is still ongoing. In the animal world, porcine epidemic diarrhea virus, which emerged 25 years ago, and slowly moved across Europe, became highly pathogenic in the early 2000s in Southeast Asia. It then spread to the U.S. in 2013. In the U.S., it killed 8 million pigs in the span of just several months. All told, the virus has killed 1 billion swine since it arrived in Southeast Asia. So the pathogenic potential and the scope of an outbreak from a highly pathogenic coronavirus is potentially enormous. And from a policy point of view, it's critical to know that there are viruses in nature that are pre-programmed to move fluidly from one species to the next. And if you know that, you need to begin to at least put some resources into developing effective drugs and vaccines. And there are no effective drugs and vaccines against SARS or MERS. And that's the situation we're in 12 years after the SARS epidemic emerged in 2003. And I think that's unacceptable.
Ralph, I want to thank you for joining us. I know you have to go now. I'm arranging a trip to Chapel Hill next year. Maybe we can sit down and talk even longer about a variety of interesting virological questions. I
look forward to that.
Thank you, Ralph.
Okay.
Bye-bye.
Kathy,
Thanks.
Rich, nice
Bye.
talking with you.
Thanks.
Bye, Ralph. Thanks.
Bye-bye.
This episode of TWiV is also sponsored by ASM Education Department. Grad students, postdocs, and early career scientists are invited to apply to attend the ASM Scientific Writing and Publishing Webinar Series. This is a 3-month, 6-part program that takes place from January through March of 2016. So it's a webinar. You can stay in the comfort of your home or office and learn great things about writing. You'll learn from ASM journal editors about the whole scientific writing and publishing process. These are people who have been involved in this for many years and know what they're talking about. And the things that will be discussed include titles and abstracts, how to write them and make them attractive, figures, figure legends, and the whole manuscript review process. So once again, it's a 3-month, 6-part series, 2 parts a month from January through March of 2016. They are taking applications for the program. You can find more about it at bit.ly/SWPI online 15, all one word, bit.ly/SWPI Online 15. We thank ASM Education for their support of TWiV. All right, Vineet.
I'll move over into the chair for the moment.
Let's circle back to that first series of experiments with the SHC 014 spike in the background of SARS. Tell us what you did there.
So, as Ralph said, we have these spikes, and our collaborators at the Wuhan Institute they've isolated a number of sequences. So I think in their original paper in 2013, they reported 18 spike sequences and a couple of full-length viruses, one SHC-014, one they called WIV1, which was the virus they were able to isolate. That virus was able to use human ACE2, and so that's the clinical isolate that they were able to actually isolate out of the bat guano, which, for those of you who have overworked with that, it's really an amazing thing that they were able to isolate that virus at all. Building from that, SHC-014 is the next virus down in terms of closeness to the epidemic strains. It's about 12% different. And when we first started looking at the strain, we had looked at the modeling, and we have a number of people in our group and other groups that have done modeling of the RBD. A lot of those studies were based on escape mutants and other interactions. The majority of the antibodies that have been generated against SARS, monoclonal antibodies that have been thought to be therapeutic or used for therapeutic purposes, focus in the receptor binding domain. And so a lot of the changes that we see there, we actually think we understand the interactions pretty well. And so when we look at SHC-014, there are 14 contact residues that are thought to interact between SARS spike and the human ACE2 molecule. Of those 14, SHC-014 has changes at 7 of those positions. And of those 14, there are 5 that have been shown to be critical for binding and species specificity. All 5 of those had been changed in SHC-014. This is in contrast to what we saw with WIV1. WIV1 only has 3 changes in those 14 residues. And of those 3, they're actually very conservative substitutions. And so we weren't particularly surprised that WIV1 could bind. We would have probably potentially predicted that. And our collaborators at Wuhan shared with us their pseudotyping data, where they'd taken the spikes and using a lentivirus system, had shown that with their SARS wild-type spike, with the WIV1 spike, that the virus could enter using human ACE2. And they showed pretty clearly that SHC-014 in the pseudotyping experiment could not. And that was, they used HKU3, which is at the other end of the group 2B spectrum in terms of the depth of the width of the coronavirus group. And so from those studies, all of the predictions are that this virus shouldn't be able to grow. And so
in human cells, you mean?
In human cells, in using human ACE2 right. at a minimum. And they'd used human, and in their pseudotyping experiments, they'd used a number of orthologs. And I can't remember if they used bat, but for what they found was they had no evidence that ACE2, bat, civet, or human could use that. It could use the ACE2 molecule. And that kind of made sense based on the fact that there are 7 amino acid changes. Now, Ralph mentioned there are changes that we see in SHC-014 that we've seen in strains that combine humans too. A couple of those mutations had been seen in civet strains, but as individuals. So of the 14, we had seen 1 change that corresponded to the SHC-014, but that's 1 out of the 7. And so there is evidence that binding could happen, but I don't know that it was or is conclusive in that way. And so when we made these viruses, it was under the premise that it was probably likely that this virus wouldn't grow. It doesn't mean that we couldn't use it for other aspects. And Ralph talked about the cartography and making antibiotic or making vaccine responses against this spike would actually be interesting because it may actually lead to antibodies that are outside of the receptor binding domain that may actually be cross-protective against the entire group. And so that was the premise of the studies. And so we built the chimeric virus with Ralph and I had a discussion on whether we thought that the virus would work or not. When I was originally doing the experiments, I wasn't thinking that it would. And so when we were able to recover virus from the viros that we transfect into, it was surprising to me. Ralph claims that he wasn't surprised. But,
So the chimeric virus you're talking about is that you're taking the SCH014 spike protein and swapping that into a mouse-adapted coronavirus, swapping the spike protein on the mouse-adapted coronavirus with this SCH014 virus spike protein, correct?
Correct. That's exactly right.
So theoretically, you're confining your attention there primarily to whether the spike protein is going to function in that context for binding and entry of a virus that is otherwise already adapted to the mouse, right?
Yeah. Yes. Okay. And I think that's an important point. When we think about the spike, a lot of the focus after the outbreak was and continue, and rightly so, is on the ability of the virus to bind to the receptor. But coronaviruses are really large. They're largest RNA viruses. There's 28 to 30 kb. And for a number of reasons, the backbone may not actually support replication in human cells itself. So this separates out whether the spike can mediate entry and whether the whole virus. So there may be aspects of the backbone of SHC014 that make it unable to grow, whether it be controlling the host immune response or some interaction with cellular machinery, that the full-length virus may not have been able to grow, but it has actually nothing to do with the spike protein. And so this controls for that in a way.
Right.
And if we had known SHC014, if we had predicted that it would grow, there's value in making the chimera as well. And we can talk about that a little bit more as we go through the data.
So why did you decide to use the mouse-adapted strain? Did you already think, wow, we could do some mouse experiments if it does grow?
Well, yes. I mean, I think, a number of reasons. Doing mouse experiments gives us a lot of information on pathogenicity. It also allows us to ask questions. If the virus does grow but doesn't grow particularly well, we can ask questions about using an SHC014-based virus as a vaccine. It also, while we don't have any hard evidence for this, in general, we do believe that adaptation to the mouse is not particularly helpful to the human. So we've done the studies where we've compared the mouse-adapted strain to the wild-type strain in human airway epithelial cells. We don't see a marked difference between them. If anything, the mouse-adapted strain is not efficiently, it's always less, but it's twofold, threefold, fivefold differences that are not virologically important. And so we don't have any reason to say that this virus is more virulent, and we don't have the data to say that it's less virulent in the adaptation away.
Yeah,
unfortunately, the only way you could really do that is maybe non-human primate studies, or I don't know that anybody's volunteering for human challenge studies, but.
So this mouse adapted strain, it's a SARS strain that was adapted to mice. Is that correct? Yeah.
So isolated SARS was taken and passaged into mice. MA15 is the name of it. It was passaged 15 times, and it gained virulence in BALB/C mice. And that is the strain that we almost always use in our mouse experiments. So in a BALB mouse, 1 times 10 to the 5th on a 10-week-old mouse will result in lethality. And that varies based on genetic background. So Black 6 mice at 10 weeks are resistant. And there are elements of this model as you age the mice. A year-old mouse is much more susceptible, much lower LD50. And so there are elements that this SARS model recapitulate both the human disease aspects and are affected by both genetic components as well as aging components.
And do you know what the genetic correlates of that adaptation are?
So there are 6 amino acid there are 6 changes. There's 1 in spike, so there's a Y436H mutation, which we think increases the affinity of the virus to the mouse receptor. It doesn't ablate interaction with the human receptor, but we do think that this virus is able to bind the mouse receptor better. And that mutation alone is not sufficient to increase pathogenicity to the full MA15 virus, but it captures a lot of that. So that mutation in spike is required for pathogenicity, but alone it is not sufficient. So there are other changes in the backbone of the virus that are required, and we've done several mouse adapted strains trying to figure out if what components and how their interaction with host components are driving this response. And so there are mutations in other locations in the backbone, and we haven't really figured out exactly what it is. So
this is the kind of experiment you could not do today, right?
Mouse adaptation of SARS would not be, I mean, it falls under the pause. So to increase in pathogenicity in a mammalian model.
Right.
Is it making it more pathogenic in humans? The answer is we don't know. And there's really no great way to test it.
Yeah.
But we also have to remember that the epidemic SARS strain passed through a lot of people and did plenty of adaptation in humans already. So we don't know if we could gain more adaptation from.
Traditionally you think of, you pass a virus in a non-human host, it attenuates it for humans. That's how you make live attenuated vaccines. So if I
I'm almost ready to volunteer for this. Stick me in a BSL-3 facility, and I'll snort some of this stuff and see what happens.
I'm afraid you can't do that, Rich. Not permitted.
No, I will mention that to Ralph.
I don't think your family would like that. It's not up to you, Rich. But if you look at all the information we've gained from studying this mouse-adapted SARS virus, it's tremendous. So and it's been around for years. It hasn't done anything. It hasn't escaped. It hasn't infected people. Why can't we use that as some data and say, this is good stuff to do and it's safe? I just don't understand why that's overlooked. I don't expect you to answer that, Vineet, but I'm just complaining.
The other interesting thing that came up is that you adapt it to one strain of mice and it's not necessarily adapted to another strain of mice. So these adaptations are really highly specific, not only just to species, but even strains and varieties.
Right.
Yeah, if Ralph was on, I'm sure he would talk about the collaborative cross. So that's for those of you who are unfamiliar, the collaborative cross is a genetically diverse mouse panel, and it takes 8 founder strains. And over the past 20 years, they've been developing mice to capture genetic diversity using these founder strains, which have 5 lab-derived strains and then 3 wild-derived strains. And while it's not equivalent to human genetic diversity, it captures the same level. And in those mice, when we put SARS in those mice, we get the full range. We have mice that are completely refractory and gain weight after infection. And we have mice that rapidly lose weight and are highly susceptible. And one of Ralph's major projects, in addition to what we're doing with these coronaviruses, is identifying host factors that drive susceptibility. Because it may not just be the virus that infects a person is important, and the genetic background of that virus is important, but the person itself and their physiological conditions and their genetics and how old they are really matter in these kind of emergence questions as well.
Yeah. Okay, Vineet, tell us what you found when you made this virus.
So we've taken the SHC-014, we made the chimeric virus, and we started with Vero cells. We found that it replicated equivalently in Vero cells, which don't have a strong immune response. And so our next step was to look in
and for those who don't know, those are monkey cells, right?
Yeah, African green monkey kidney cells.
Okay.
And then we went to respiratory cells. Calu-3 cells are a cell line that we've used. We use the 2B4 version of these. These have been sorted for ACE2 expression. And
they're human, right?
They're human cells. Type 2 alveolar cells are closest to alveolar cells. And then primary human airway culture. So these are derived from lung transplant donors. So the upper branches of the airway are available. They're not always used. And so a researcher here in the cystic fibrosis core, Scott Rendell, harvests these and is able to make cultures, and takes weeks to make these. And he's been doing it for several years, where you plate the cells and you can actually differentiate them into a ciliated airway, a primary human airway. And so they're grown on air-liquid interface. So You have an insert. The top portion is exposed to the air. You can actually see the cilia, and they beat. And the lower layer has the basolateral media. And so they mimic the human airway as well as any cell culture model could. And we've done a number of studies with SARS over the years with these cultures. And we started with those with the SHC014 in the SARS backbone and compared it directly to the epidemic strain, the epidemic Urbani strain, and found that it could replicate equivalently to the epidemic strain. And from there, we took that same virus and infected mice. This is 10-week-old BALB C mice, and found that it was not as pathogenic as the mouse-adapted MA15. So those mice challenged with 1 × 10⁴ dose die between 4 and 6 days post-infection. The SHC014 mouse-adapted virus, the SARS backbone virus, caused about 10% weight loss in BALB C mice. Significant disease, but not particularly lethal. What was interesting to us was that the titers were roughly equivalent 2 days post-infection between the viruses, and it appeared that the virus was able to carry out a lot of the same program that the mouse adaptive strain could. And so we had this virus that the full-length the spike had mediated ability to replicate in human cells, and while it didn't cause equivalent disease, it was causing disease in mice. It wasn't equivalent to the mouse-adapted strain, but it was definitely not attenuated.
Right. But it's, it has reduced virulence in that. Whether you can use that as any kind of comfort, I don't know, because mice are not people, as we often say on this show. But like in many cases, when you make artificial viruses, and I would say this is an artificial virus, so we don't know if it exists out there, you interfere with the ability of the virus to cause disease. So yes. I guess it's not surprising, right?
Yeah. And
so let me make sure I understand this. The hybrid virus with the spike-glycoprotein from the bat in the mouse-adapted background.
Not bat, but bat virus.
Bat virus, sorry. Is not quite the same as the mouse-adapted virus itself in animals, correct?
Right. It's not at all. And in a lot of ways, and I think there's actually a lot of interesting aspects of that, and we didn't really have an opportunity to expand on this, but one of the observations, it's in the paper, but we didn't go into length about this, was we actually see a tropism difference between the SHC014 and the epidemic strain, the mouse-adapted epidemic strain. So the MA15, the mouse-adapted SARS wild type, infects both the airway and the parenchyma when you look at the lung sections. It's very clear the virus is in all areas of the lung. In contrast, SHC014 in the SARS backbone only is replicating or only stains in the parenchyma, and the airways are largely absent. The large airways are largely absent of any viral antigen, which is really interesting in terms of pathogenesis and exposure. And considering that our titers were very similar, it argues that there may be actual tropism differences. And these tropism differences may have to do with protease cleavage and the ability to infect those cells. And so it's actually something we're following up on now. We've been looking at host proteases that may be differential between the two in those locations. And so it actually argues that one of the reasons that SHC014 may be attenuated relative to the mouse-adaptive strain is that it doesn't infect these airway cells, or these large airways are not getting infected. It's a real opportunity, I think, and something we're really interested in. So much of the focus on the spike protein, and rightly so, has been focused on the ability to bind. So the receptor binding domain is where a lot of the focus has been really focused on this RBD region, and there's a lot of difference in this region. Less focus has been focused on the protease cleavage site and the importance of that in emergence. And one of the things that this virus actually gives us an opportunity to, we can actually look at this attenuation and ask, are we going to be able to attenuate the epidemic strain, the mouse-adapted epidemic strain, by doing a domain swap. If we take the receptor binding domain of SHC014 and put it into the SARS backbone, will that reduce its ability to replicate? Or if we take a chunk of the protease cleavage site and find that it's not as efficient, that may be a critical step in emergence that we've never even considered before. And we couldn't have done without a virus that actually binds to human ACE2. And it's something that we're pursuing now. We're asking these kind of questions that while the SHC014 virus is able to grow, it's not equivalent to SARS, and that can actually inform how the epidemic became an epidemic. I mean, I think we're still very early in this response, but there are lots of tools here in telling us more about how these viruses emerge by going from the bat to the human and comparing them between the two.
Although it's logical, I still find it fascinating that subtle changes in the spike protein could affect what tissues in the host are infected and thereby affect pathogenesis. That's really interesting.
Kathy.
I was just going to say that I think in this figure where you do show that histopathology, it's a really nice, convincing figure about the difference in tropism with the 014 strain compared to the epidemic strain. And I understand that at some point, something on the order of 2,000 words had to be cut from this manuscript. And so you show this one figure about tropism difference. Are there other things, that are going to end up coming out somewhere else in terms of this that you could mention at this point?
So
differences between the two viruses?
Yes. So I don't know how much I should say. So this is work that your former student Lisa Gerlinski is working on. Mm-hmm. We have a number of mice that have protease. We have protease knockout mice, human host protease knockouts, and we have one where the epidemic mouse adapted strain MA15. There's no phenotype. It looks exactly the same as the wild type, and that the SHC014 virus, which causes about 10% weight loss, has no replication, and is very low. So we think that protease may be critical. Mm-hmm. I'll let Lisa, if you want to talk to Lisa, she can tell you what.
Sure.
What it is.
And the what you mentioned about the collaborative cross. I know you're a co-author on this paper that's in PLOS Genetics that maybe we can stick in the show notes. It's one of the cases where the collaborative cross has been used for infectious virus, looking at genetic differences in disease susceptibility. It's one that's being cited now. So it's open access, and we can show that.
Yeah, can you stick that in the show notes?
It's already there.
Very good. All right, Vineet, can you what do we do next? Let's move.
So we've built at this point we've built the chimeric virus. At that point once we knew that it was viable, we'd ordered and we'd started process of getting the full length viruses made. Obviously, making the SARS, making the spike is not trivial in its length. Making a full 28 kb genome is even less. It takes time. And so we initiated studies asking questions about the therapeutics that we had available to us. And so we think vaccines and we think monoclonal antibodies. And so the monoclonal antibodies, we've had a long-standing collaboration with Wayne Marasco's group at Harvard and then Antonio Lancevecchia's group, making antibodies for SARS. And then a number of studies that we've done over the years where they've given us antibodies and we've created escape mutants, and then they've made new antibodies against the escape mutants. And the idea was that we could drive an antibody response, make a panel of antibodies that could really block anything out there. And we spent a lot of time making chimeric viruses with civet strains and diverse strains, trying to figure out ways to improve this antibody and its breadth. And so with SHC014 and how different it was in the receptor binding domain, we saw an opportunity to ask the question, well, do the antibodies that we would pick, and the 4 antibodies that are in the paper, are what we would have used if we were designing a ZMAP, the ZMAP equivalent for SARS, these are the 4 antibodies that we would have picked. And so in vitro, we took these antibodies and we asked whether they were able to neutralize SARS, which it does, and SHC014. And in all of them, the neutralization was weak at best. 3 of them were outright failures that we didn't get over 50% neutralization. One of them approaches 50%, but you would have to use in a human grams of antibody. And part of the reason is, and we've mapped a lot of these antibodies, we know exactly where they bind. Almost all of them are binding within the receptor binding domain. And why did we make antibodies that bind the receptor binding domain? Well, they are highly neutralizing, and they're the strongest of them. And for the most part, the expectation was the viruses that are SARS-like that can bind human ACE2 wouldn't tolerate major changes in the ACE2 binding region. And here we find very clearly like that SHC014 had a very different RBD and could escape these antibodies because the residues are different. And whether the tract of the binding is slightly different, and that's the difference that we see, whatever the reason that these antibodies don't bind, despite the ability of this virus to replicate, it really clearly showed that our antibody responses, what we've developed, are not sufficient. And I think what's important is that Lancevecchia's group and Rasko's group, they have a number of antibodies that we have tested in the initial phases, but we kind of went with what worked. And we have an opportunity to go back and ask questions on, to go back to those antibody stocks, the ones that maybe we didn't go through first, and ask which ones are there, which ones can we expand upon and look at with this SHC014 virus, and actually maybe find a better treatment model. And it may be just adding another antibody that has more broad response. Unfortunately, we couldn't have done this without this SHC virus that's kind of, as Ralph talked about earlier, the bookends. Without a virus that was in the middle, we really didn't have an opportunity to do that. We needed something that was different enough to really chase down antibodies that are outside of the RBD.
So this result scares a lot of people, but you need to know this, right? It's like Ralph said, you're going to wait for something to arise that you can't treat with monoclonals, or you're going to be proactive. And here, this shows you that there's spike genes in bats for SARS-like viruses circulating that are really different. And this tells us we should try, as you say, make something more broadly neutralizing, right? And
I think, and Ralph's right about that. SHC014 may never emerge. But but we can improve the cocktail with this. And it's not that we don't have these resources. It's just that if you had asked me whether the antibodies would work, I mean, I don't want to appear that I'm always wrong, but consistently in this paper, I really thought one of these 4 antibodies would have worked. And I was surprised that one of them didn't work. And, I think often some of the most interesting science is when you're wrong. But I mean, I think this is an opportunity. I think this is why we want to do it. We are in better position to deal with a Group 2B coronavirus because of this SHC014 virus. And moving forward, there's a lot of opportunity to learn about emergence and therapeutics from that information.
Now I'd rather be scared than ignorant.
Yes. So I think this is a good point. You have to take some risk when you do science. Anything that doesn't have risk is not going to be very good all the time. So I think that, you mitigate the risk and you do the experiments. I just think the people who criticize this don't understand. They don't, as Ralph said, they don't know the whole field of coronaviruses, and it makes it hard for them to realize what's going on. And I recognize that that's an issue, and I think they should listen to this episode. They won't. That's the problem. Vineet, then you did some vaccination studies, right, with this virus?
Yeah, so we have lots of different approaches for vaccination. In the field, a lot of effort was made to make an inactivated vaccine. This is a SARS particle that was doubly inactivated. I think we call it DIV in the manuscript. And it had been shown to be effective in young mice. And then our group had gone back and done this in older mice, year-old mice, and found that there was immune pathology. And as the mice got older, the vaccine responses waned and actually were partially pathogenic. It was actually worse to have this vaccine than to not be vaccinated. Another aspect was that if you challenged the virus that was heterologously different, you amplified those immune pathology responses. And so the DIV vaccine itself was not particularly well received after that point, and isn't really a viable candidate moving forward. But we tested the ability of that because it is a full-length SARS virus, and we wondered if this DIV vaccine could protect against this SHC014 virus. And what we found in both young and old mice is that there was no protection offered by the live attenuated vaccine, which was not particularly surprising. But we would have expected we could have expected some neutralization. I guess this is an example of we didn't think it was going to work, and I was actually proven correct. The other aspect was we did the inverse experiment. So we have a number of live attenuated vaccine platforms that work, whether we knock out particular aspects of the virus or efficiency of the virus. We knock out different components. Our fidelity mutation virus has been used before as a vaccine platform. Luis and Juanes has a delta E protein deletion that he's used as a vaccine platform. And we wanted to ask if we took SHC014 and we infected with a dose that was that caused disease but wasn't lethal, and that's the maximum dose, could we offer protection? And what was interesting was we did see protection. If we infected the mice, year-old mice, with a dose that caused 10% weight loss, when we came back at 28 days and challenged with the lethal MA15 challenge, those mice were completely protected. And so SHC014, the spike components of SHC014 are able to confer protection against the SARS infection. There are overlapping epitopes between these spike proteins. When we looked at the actual level of the sera, what we found was that the neutralization is at 1 to 200, so the dilution is very low. I mean, we need a lot of that sera to neutralize. For comparative sake, our vaccine platforms are 1 to 1600. And so the expectation was that even though this vaccine works in the short term, long-term immunity is probably not established. And this tells us a couple things. One, there are common epitopes between this SHC014 spike and the SARS spike that we may be able to manipulate when we make antibiotics or antibodies to this region, that they may actually have cross-protection opportunities. I think the other important aspect is when we went back and we challenged the mice with a lower dose, a dose that didn't cause weight loss, this was 100 PFU, this actually didn't offer any protection. And so it signals that this spike protein, that there is overlap between the SHC014 spike and the SARS spike, that we can drive protection, but this may be an issue that the major antigen long-term, it may be really difficult to tease this out. And so we may have to design new approaches to actually pick for broadly neutralizing antibodies as opposed to ones that are specific to the receptor binding domain, which is not common between SHC014 and the SARS spike.
All right. So after all this was done, your bat coronavirus was ready, right?
Yeah. So,
so I wanted to. There's a practical issue here. Is the initial swap of the spike glycoprotein into the mouse adapted background. That's the logic of doing that rather than going for the full length virus to start with is just that it's a lot easier, and if it didn't work, there's no point in going any further. Is that the thinking?
So I mean, there are a number of reasons. One, you have to remember, based on the evidence and the data, this is a virus that shouldn't have grown.
Right.
And so you have the practical aspects of you can make the spike protein and prove in the mouse, in the chimeric virus, that doesn't grow. And so that's both the fastest and the cheapest option. So
and then you're done, and you don't have to make the whole virus, right?
Yeah.
Well, and the other aspect is, even without that, if we known the full-length virus could grow, there are a number of reasons that it wouldn't grow that have nothing to do with spike itself. So those background changes in NSP9 or other components that are adapted, you may actually have a compatibility issue with the host, that either the immune response stops it or some interaction with some cellular machinery is required. And this virus may not grow, but it actually has nothing to do with binding and entry. It actually has to do with replication or the immune response.
Okay, so the constructs are really complementary in that regard.
Well, and I think, if we had known that SHC014, the full-length virus, could replicate, it's likely we still would have asked about the chimera, because the chimera tells us about the spike itself and isolates the difference of the spike. And as Ralph said, these pools exist, and recombination is well established in coronaviruses, and in data that we've seen from our collaborators at the Wuhan Institute of Virology, there's tons of there are recombination hotspots in the spike, and they have all these spike sequences that they've compared, and they actually see regions where this is directly cut from another spike, and the only way this could happen is through recombination. So I understand. I mean, this gets into the hindsight of the results. If you know that the SHC014 virus is going to replicate, we would probably start with that. At the same time, we didn't know that at the front. And the second is the chimera still has value in these responses.
Right. Okay.
Also, it's a huge resource expenditure to do that. And like you said, it took time, whereas you could do this, it sounds like.
So, it took us.
Chimera much quicker.
Yeah, I mean, you're talking about, 1,200, 1,300 base pairs versus 28 KB and a bunch of different constructs. You're talking a month versus a couple of months.
I have a question about the fact that you did this, made this full-length virus, and you compared it to the SARS epidemic strain virus in mice. So I thought that you had to mouse adapt the virus. So what can you do with the viruses without mouse adaptation?
So we are comparing, in the mice, we compare to the mouse adapted strain. In the human cells, we compare to the epidemic strain.
I guess maybe I misinterpreted the figure then. I thought that you just put the non-mouse adapted virus in and the non-mouse adapted full-length 014 in.
It looks to me like in 3D you got all three. You got Urbani,
right?
You got the reconstructed bat virus, and you've got the mouse adapted virus, if I'm reading that correctly.
Right.
So you're comparing all three.
Right. Okay.
So your question is, why does the bat virus replicate in mice without adaptation? But it's not the human virus, so maybe that. Vineet, if you just take human SARS isolate like Urbani and you put it intranasally into mice, BALB-C, does it replicate at all?
Yes, it replicates with roughly equivalent. The titers are lower than the mouse adapted strain, but there's a large amount of virus in there.
Okay.
And we have a number of research. We have more depth in this question as well. We have a mouse that expresses human ACE2 under a Clara cell promoter, and we've done comparisons between the epidemic strain, the Urbani, with no mouse adaptation, to the SHC014, and we have interesting data there that shows clear attenuation, but there's complicating factors of that mouse is not as clean as we would hope in terms of where it's expressed.
So, but the epidemic strain, the Urbani strain, infects the mice, but it doesn't kill them. Doesn't look like it even makes them very sick, right?
No. So you're talking about in Figure 3. I had to pull up the paper because
right.
So Figure 3. So Figure 3 is the full length SHC014 versus the epidemic SARS. So
Right.
I didn't include,
right,
the Urbani. And so the expectation, the wild type MA15, which isn't included in the paper, but those mice do die completely. So looking at the paper, it's the gray line is mouse adapted.
Right.
The green line is the full length SHC virus. The black line is epidemic SARS.
Right.
Not mouse adapted.
So the bat isolate SHC014 replicates in vero cells. It replicates in human airway epithelial cells, and in mice, it causes more weight loss than does Urbani or the mouse adapted strain, right?
No. So the SHC full length doesn't cause.
Oh, that's the gray is the mouse adapted. Sorry.
Right.
So then the full length virus it grows equivalently in vero cells, is attenuated in primary human airway cells.
Got it.
And is attenuated in mice in terms of replication. Neither the Urbani strain nor the full length SHC strain cause any weight loss or pathogenicity.
Got it. Yeah, I have a problem with gray versus black here. I can't see.
Yeah, it's subtle. Okay.
All right. So,
All right.
So that's the full-length bat SARS-related coronavirus. You made the entire genome as DNA, and then you rescued virus out of cells.
Right.
And then you used that to do these experiments.
Yep.
And also, another thing that comes out of the same mouse experiments is that the full-length bat virus does not replicate as its pathogenicity in terms of weight loss in the mice is the same as the epidemic Urbani strain, but in terms of replication in the lungs, the full-length bat virus doesn't replicate as well as the Urbani strain. Correct. So by a couple of different criteria, replication in human airway epithelial cells and replication in the mouse lungs, it is not as robust an infection as the epidemic strain, correct?
Correct.
Okay.
So, Vineet, I've been on Twitter all week defending this paper.
Yes, I saw.
Because I know you need to do experiments, so I'm happy to do that. And at one point, I got in an argument with someone who said, why was this recombinant virus made? Why didn't they just study the bat virus? I said, they did. It's in the paper. And, he said eventually, sorry. But I think it just shows that most people don't even read the paper. And they just read either some newspaper article or some online article, and then they make a decision based on that. And, I bet, so two people have been quoted widely in the press, Simon Wayne Hobson and Richard E. Bright, and they have been notable anti-gain-of-function advocates, and they're quoted negative sayings. And I would bet, I could be wrong, but I would bet they have not read this whole paper as much as we had, and therefore don't appreciate what's in it. And I think this is really bad because this is driving the whole argument that people that don't read the papers are participating. Does that drive you crazy?
It's, I think there's a certain amount of knee-jerk reaction to anything that's gain of function. And I think it's important to consider, and Ralph and I have talked about this, there is a fundamental difference between pursuit of gain and being prepared for gain. I don't think anyone would argue that we expected this virus to be more pathogenic than the epidemic virus, and it hasn't been. I think being prepared for that possibility is important, and I think it's a consideration that we have to consider. But this is very different than mouse adaptions or transmission studies where the gain is what we're seeking. Here, we don't know anything about these spike proteins. And it's very easy to be paranoid about the gain of function aspect to this and being prepared for it. And we really don't want that to be captured in any kind of rules that, well, we don't know what's going to happen, so you shouldn't do it. I think you could argue that any adaptation of SARS, any manipulation of SARS could result in a gain, so we shouldn't work with SARS. Is the down, that's the logic goes down that way, that, oh, well, you added GFP to SARS.
Yeah.
Well, that could lead to, it ignores the data that's available and ignores scientific insight. And we have been rightly proven wrong before, but I guess that gets to, the stuff that we have in place. And I think it's hard, as Kathy mentioned, we cut a number of words from this manuscript, and I'm not even sure that we would have covered the things that people are interested in. So, and I think this is a great opportunity to talk about the process that we went through to come up with these viruses. And we've spent a lot of time figuring out the best way to get at these questions. And so we had local approval. We asked about these viruses, putting them in the backbone. We had the pause. We had these interactions with NIH. And I think there was an aspect, and Ralph talked about it, that there's a black box when this stuff goes out. And we don't know how it's reviewed or who's reviewing it. And we made the point that when we sent our materials to them, that we didn't think that this was a gain. It could have been a gain. And if we'd had a gain, we specifically have criteria for what we define as a gain and what we would do. And
Ralph described that preparation of who you would notify and, then what would happen. So it was clear that you were prepared for that.
Well, and I think, Ralph went into, and we've talked about these studies, it's really hard to talk about, there's not a place in the paper to talk about what we do and how these countermeasures are in place. I think, we hear about these safety breaks and all of that, but we don't know. Each individual lab has its own protocols. We actually have gone through and we've counted the number of things that our lab does in terms of safety. And so we have 9 levels of PPE that we use in the containment lab. In our lab operating procedures, we have 31 different things that we do in terms of our protocols. And lots of these things are overlapping elements that if you don't do one, you should be captured by 2 or 3 of the other things. Outside of the lab, in terms of, if we have a cold or something like that, we do drills. We have another 18 things. So you have all of these layers because, there are accidents, there are mistakes, there is human error. You can't exclude that. No one is perfect, and expecting perfection is problematic. But we spent a lot of time designing this approach, and I think that's often in the gain of function criticism ignored, that I think it's very easy to think about the movies, and you have scientists are on their one end of the spectrum. You're either the crazy scientist that never considered the ramifications of their work and made something that will destroy the world, or you're the lone scientist that saves the world with your cure. And there's no in between. There's no gray in the movies. And so often there's no gray applied to us. But I mean, we know what we're working with. We know probably better than anybody what the risks are. And I think Ralph talked about it. We don't want to be in a situation where a coronavirus emerges and we think we could have done something about that, or we could have been more prepared. At the same time, we know that, is this a risk? Yes. What do we need to do to mitigate this risk? We put all of those things into place. We consider all of these things. I think to imply that there's arrogance and we just want to do this because we want to do it is really painful. And I think, scientists have to be aware of what they say, because, what you say when you have a position at a university or you're a researcher can be misconstrued and taken out of context or used in ways that you wouldn't really think. You can justifiably, I think we can have differences of opinions, but it's important that we consider the impact of those that one sentence can be taken out of context. And I think, in our discussions, it's important to consider that critics and proponents on both ends of the spectrum have to consider that oftentimes you're getting non-scientists or people who are reporting it that may not fully understand the width of the statement.
I think this is so right on, because, you can't expect people to read your paper. That's fine. I think it's great to have a discussion, but, the anti-GOF people are simply negative. You cannot do this. You're going to kill the world. That's not a discussion, right? That's just saying you can't do these experiments. And I think the key here is, we try and provide a forum for virology here on TWiV. You can talk for an hour and a half about your work. You're not going to get that anywhere else in the world. No radio station, no interview, nothing. Not even a talk at ASV goes that long. and it's not back and forth. So I think this is important, but unfortunately, Wayne Hobson and Ebert will not listen to this and get a real appreciation of really the amazing things we've learned from this work. But the other key here, I think, and part of the problem is the press release from UNC was really bad. I don't know.
Yeah, I thought they kind of blew that.
The headline was picked up directly by several websites, and they say something like, investigators show that bat viruses can go right into people. And of course, I don't think you'd agree that that's correct. And this is a problem. And I know we've talked about this a lot on TWiV, and I wish Alan were here. But often press releases get the wrong message, and investigators are not supposed to be writing them, but they should. I don't know. It should be more closely involved. So I think that's part of the problem here as well, right?
I don't disagree. I think part of it is the press release. I mean, it gets to, we have, the press office at the university has a different purpose than the researcher.
Yeah.
You may argue it is, when Ralph and I had a discussion, is it inaccurate what they said? I mean, you could argue that they didn't say anything that wasn't true. Is it inflammatory? Absolutely. Is that what brings eyes to the paper? Yes. I mean, I think fundamentally when we looked at that, if you read the headline and took that and just took the headline, it's bad. If you go into the paper, Ralph talks in the press release about, we're further than you think, but like there's important aspects, but this virus, there is aspects in the actual text that minimize that headline. But if you're taking it with the context that you want to, if you want eyes, the headline did not. The headline drove that, and it probably drove a lot of the negative press too.
Yeah, the Daily Mail took it. New SARS-like virus could jump directly from bats to humans without mutating.
I think they could have come up with a headline and a lead line for the article that were equally catchy but not so inflammatory. I wanted, because you talked about the biosafety and biosecurity stuff, there's almost a full column of discussion of it in the online methods, which again, if people don't read the paper, they're not going to recognize some of the things, and you've even elaborated some more of them. I wanted to know whether in submitting this paper, you had to go through any additional hoops, or was it simply submitted to Nature Medicine?
So we submitted it with paperwork from our final approval letter from the NIH. So the kinetics of the approvals went such that we initially applied for approval to do the work in the spring of 2014 to make the viruses. When the pause went into effect, this project was not under the stop order that the MERS mouse adaptation was. So we had until the roll date of the grant to respond. And we sent our paperwork where NIH asked for, our process. And this grant covers a number of, it's a large collaborative grant that has Ebola and other groups in it. And so there was a number of things that we had to answer. And at the end in May, before the final approval, we basically got asked for a letter saying whether we could do these experiments, continue these experiments or not. And we actually submitted that paperwork to the journal with the understanding that they wanted it.
That was before it even went out for review. Is that correct? Did you get any?
Yes.
Was there any comment from the reviewers that required extra action?
We did it on the front end, just so that it would be available to the editors and to the reviewers as needed. So we were proactive in that sense. We expected that this would be a question, and had been. I think if we hadn't included it, people would have asked.
Okay.
Sure. Mm-hmm.
Well, I do hope, and Ralph mentioned this, the people who are making these decisions about the future of these gain of function with flu and SARS viruses, I hope that they look at the science and really disregard all the hype. They disregard Ebert and Wayne Hobson, everything they say in the press. Because if you look at the science, you understand that this is important work. And I'm afraid that this is going to be lost in the shuffle, and science is gonna lose out in the end. I'm afraid of that, but I just don't know what's going on, because as he said, we don't even know who's making this decision.
Well, and I think, I don't want to characterize. I mean, while we don't know the NIH process, they have been very supportive of this work.
Great, that's great.
They've been very complimentary, and in a lot of ways, it's important to realize that while NIH is administrating they're at the front lines of this. The policy came down from the State Department. The pause is not an NIH directive.
Right. Right.
And so at a certain point, they're tasked with applying metrics that are not necessarily scientific, that may be more political than anything. And so they're in as difficult as a position as we are. And we're all operating, I think everybody I've interacted with on this, at the NIH and with oversight, we're doing our best, and we want to minimize any risk that there is. Implications that, it's really, I expect the Daily Mail, or I actually showed Ralph the Daily Sheeple, which was, I think so. We kind of laugh at these. You kind of expect that. The internet is what it is. I think it's really disheartening when you have scientists who have the opportunity and can look at the paper and justly look at it, making these comments. And as you've said, I don't know how in depth they've looked at it, but it's been said, words matter, especially when you're in positions of power. And so I would encourage critics and proponents to really consider that when you make comments, that you have to have a full understanding. And if you don't fully understand, and I think there's no problem in asking and having this discussion. I had Twitter discussions about this. I've talked to a number of people, and was I surprised that it happened this way? I think in the culture that we live in, I think part of this was expected. But dialogue is really important. And it's important that we talk with people and get their feelings. I really value scientists that engage with me on this, because I have my own biases. I think this work is really important, and I don't know that, part of me thinks that it's probably good that I'm not making the final, that NIH, it makes it easy that NIH makes this final decision, because I have real interest in this one way or the other. Now, that being said, I have lots of ideas and lots of interesting things that I can follow up on. It's important that this work be done. But if society or scientific society thinks that it's too risky, I can understand that.
Well, if you have a good discussion and that's the outcome, that's fine. But Ebright says the only impact of this work is the creation of a new non-natural risk is absurd. It's patently absurd to say that. He can't have read the paper, right? This is what makes me crazy. But you're very level-headed, Vineet. So I'm glad that you're doing this work, and I do think that people need to talk about this. And I'd like to have Wayne Hobson on TWiV and talk about it. And he's supposedly a smart guy. Let him tell us why he's really concerned, and we can go over it. I think we have the best forum for that here to do that.
We've done several of these episodes where we've gone over a paper that is controversial in this regard. And I have to say that for me personally, every time we do this and go through an episode, when we're done, I come away thinking, first of all, that there's not nearly the danger or none that people make a flap about. And second, I put myself in the position of how much I've learned from this paper, and what I would miss if we didn't have this work. And it's amazing, okay, because you learn so much from this stuff, and without it, there's this vacuum. It doesn't make any sense.
Mm-hmm.
Right. Kathy, what's your schedule? You need to leave?
No, I'm fine.
You okay?
I'm just gonna skip the.
But you have a flight to leave at 1:30.
Yeah, that's at 1:15.
We'll be done by then.
Yeah, I hope so.
But I think.
I just want to add. I think it's important. And I think part of it has been, we've had a lot of questions, and Ralph gets questions about the transmissible flu studies, and it's really hard to comment on them because we work with respiratory viruses. There are similarities between SARS and flu, but the ins and outs of flu are difficult. Understanding all the literature and all that, and these people who are working in these areas really know these areas really well. And so, my wife is an educator, so she's a teacher, and I said that it's kind of like asking a kindergarten teacher what to do with senior level English class. It's not that they can't. It's that, and like a really good, I can comment on flu, but I don't have the experience or the understanding that someone who has all the experience and understanding would. And I think it's important. And I think often we look at these groups and say, oh, well, you guys are biased. You guys are working on the literature.
Exactly.
But at the same time, it's hard to find people that aren't biased in some way to comment on it and who have I read these journals, and honestly, if I wasn't working on this paper, I may have looked at the figures. I don't know that I would have I would have read the abstract. I would have looked at the figures, and then I would have moved on. It's hard to make strong statements one way or the other, but I think it's also important. My opinions, my thoughts are often discounted by that gain of function group because I'm right there. I do have a conflict, rightly, but at the same time, having a conflict it's also how people say, oh, well, these people who study climate change have a conflict, so they can't talk. It's kind of a ridiculous.
Who's better, as you say, who's better qualified? But so the gain of function opponents, some of them are signed. Wayne Hobson's a virologist, but he may not have read these. And so who's going to make the decision? That's what I'm concerned about. And, to circle back to something you said earlier, these newspaper online articles don't matter too much. But you know what? I think Congress sees them. Unfortunately, I think their staff gives it to them, and that informs their opinion, and it's really unfortunate. And I don't know how to combat that. It's a big communication issue for sure.
Yeah, and I think, it gets back to there are a number of things that the gain of function arguments drew clear lines. And it was interesting seeing, I could have predicted, I think we all know who the critics are and who would have been quoted in these stories, I think.
Yep.
And it gets to, they point out a conflict for me or Ralph in these studies, but if you're on record as being against gain of function 8, 10, 20 times, if that's your claim to fame, aren't you also in conflict? I think that is not considered when, I don't know Dr. Wayne Hobson at all. I'm not familiar with what his scientific work is, but I do know he's a gain of function critic, and if that's what you're known for, is that relevant? I don't know. But I think
I agree. I think that they have an agenda. They just don't like the idea of this. And so they will not even read the paper to understand it. They would say, no, you shouldn't be doing these experiments, period. And the public says, okay, this is a scientist, this is a virologist, so it must mean something. And but to their credit, some of these articles did quote Ralph, which is good. They have both sides, which you're supposed to do. But of course, the headlines don't reflect that at all.
Well, and I think in Ralph, Ralph and I have talked about it. You have these interviews that can be long, but
they take a sound bite out of it.
Yep.
Well, and there's, I think in having this platform is great for it. There's a lot of nuance to this. If you don't know anything about viral, like the background behind it, why didn't we make the full-length virus to start with? Is a reasonable question based on that, and I think we've talked about why we started with the chimera here, but there aren't great platforms for explaining that.
I think TWiV is a great platform,
and I am happy to chat about this whenever you like.
I just don't know why more people don't listen. I realize we're long, but. And I have a bias, but I think it's founded in good science. But there's nowhere else, as I've said, where people could talk in depth about this and get the real story, right? Am I wrong about that, anyone?
I think it's good to talk in depth. I'm just going to go out and say that I'm probably a Pollyanna about this, but I really hope that Simon Wade Hobson and Richard E. Bright read this paper. If they didn't, then they really don't have any business talking about this. As scientists, they should do that.
Not just read it, study it.
Oh, I doubt it.
Well,
I understand your position, but I doubt it. From what he said, how can you say this doesn't give us any information? We've just talked about this for an hour and a half of all the cool stuff we've learned, right?
I see your point, and I agree. But they can read it, and like Rich said, if they just read it and didn't study it, if they just read the abstract and looked at the figures and read the press release, it's not enough.
Yeah, I agree. That's what we're trying to do here, is to supply people with a digestible form of the information. I wrote a blog post yesterday where I tried to make it simple for people.
It's a really good blog post. People should go to it.
But
virology.ws.
I just don't think people will read it. People will not listen. We have a lot of great listeners and readers, but not enough to inform this this whole debate, I'm afraid. I think I would love to dedicate this episode to just this one topic. I don't want to. We had another topic planned, but we'll put that for another time. Vineet, how much more time do you have in the Barrack Lab? What's your plan?
Well, I am, as Ralph has told me, I am transitioning. So he is. He's like, "Oh, weren't you on that email?" And I'll be like, "I wasn't on that email for the group for that meeting." And he's like, "Oh, I am transitioning you out. I keep forgetting that." So I am currently in the job search mode, and so I expect I will be here. I work on this stuff, and as I mentioned earlier, there's lots of interesting host components about genetic, the CC, and having how much is driven by the host genetics and the aging aspects of coronavirus disease. I think what's interesting is a virus that is circulating in bats, it just as important is that it actually interact with the right human for emergence. The genetic components of the human can drive it, and the age of the person that's there. Coronaviruses, SARS and MERS, the lethality goes way up as you get older. And so susceptibility and changes in our host responses as we get older are really driving these responses. And that's one of the things I'm really interested in, is finding the host factors both in the genetic component, the genetic side, and I think Kathy mentioned it in Lisa's PLOS Genetics paper. She really drives into the genetics aspects, and I've got some funding for doing aging work, looking at aging in mice and asking why susceptibility is changing as the mice get older, and hopefully we can apply that to human disease.
Very interesting. So do you want to have an academic position? Is that your goal?
That is the plan at the moment.
And
I think I want an academic position.
And you'll continue coronavirus work, right?
Yeah, primarily using coronavirus work, using coronaviruses as a platform. But I think relating it to flu and other viruses is really a great opportunity to interact with other groups. Really driving our understanding of. I think often we've worked in silos of our own individual viruses, but comparing and work we've done with Yoshi Kaoka's group, comparing SARS and high-path flu and MERS, has really given us real great insights into how the host is controlling viruses globally and how different viruses use similar approaches to drive these different responses. And so I think there's great virology there. There's great interest in host immunity. And of course, we have these emerging viruses that are really interesting too.
Where did you do your PhD in? Was it virology?
Yeah, so I went to Washington University in St. Louis. I was in the immunology program there. I came in through, and I worked with David Lieb on herpes simplex virus, working on how, looking at host components that were driving differential disease. I focused on, there was an innate component that people had used. People are familiar with IRF3 and IRF7 and the interferon responses. And so I was looking at how different tissues have different requirements. So the brain really requires IRF3 to control the encephalitis portion. And without IRF3, you could have control at peripheral locations. But once it got to the brain really needed this internal IRF3 more so than the systemic responses. And so that's kind of where I jumped into looking at. I was interested primarily in the immunity. I think, I operate on, if I'm in a group of virologists, I say I'm an immunologist. If I'm with a group of immunologists, I say I'm a virologist.
That's good. Well, Matt Freeman told me that you would be a great spokesperson for this paper, and he was absolutely right. You're really articulate and calm and level-headed, and I'm looking forward to a great career in virology from you.
Well, I appreciate that.
I was thinking the same thing. Great spokesman for the paper.
Well, I appreciate that, and that's the nicest thing I think I've ever heard from Matt about me.
Yeah, he's a tough guy.
He also told me good things about you in person yesterday, so yeah.
I imagine that our relationship is that we would never say the nice things that we think about those people to them. Matt's a great person, and he's really helped me in understanding how the transition is working. And I think what's underestimated, and I think Julie Pfeiffer kind of mentioned it about, in all of these gain of function stuff, one of the conversations I have with a lot of people that work with me, with Matt, with Tim Sheehan, who's in our lab, other young people, is it really worth this effort? We all want to do really good science, and we all have lots of ideas. Are we better off doing this with MHV and not doing it with SARS? Because, it's a consideration. I think I worry, Matt's talked about this with me. You make a mistake in your BSL-3, an innocuous mistake, and you're shut down.
Yeah.
So am I taking my career? Am I taking a huge career risk? It's very easy for me to do a lot of this work with the umbrella of knowing how well established Ralph is and how respected he is, and having these in place. But as a junior investigator, I have to ask myself the question: is it worth the effort? Is my department going to stand behind me if I make a mistake, or if there's a problem, or if there's controversy? I think UNC is committed to Ralph in a way that he can go out and make have these discussions and do these kind of works. It's important to remember that this kind of work and the criticism and all of that impacts people. I think it goes down the line. Young scientists we really do think about these things and how they affect our career, for bad and for good.
That's a great point. And this is something that Julie Pfeiffer brought up originally with the moratorium, right? She wrote a little piece in MBio about how it's affecting the future scientists. And of course, you need to find a department that will support you. And when you interview, you got to bring that up. This is tricky stuff, and you're going to support me. I tell you, Vineet, if I were a chair and I hired you, I'd give you 100% support for this. But I'm not, so that doesn't do you any good. Okay. So it just makes you feel good. But that's a great point. It really is. Now, let me do a few administrative things here. I want to read just a couple of things, and then maybe we could do some picks and wrap it up. Does that sound like a plan?
Mm-hmm.
Sure.
So the reason why we did this paper at all is I got an email, I think early in the week, from Emily, who is a graduate student at UNC in the Da Silva and Barrick laboratories. You probably know Emily, Vineet, right?
I do know Emily.
And she wrote, TWiV hosts, ! I want to suggest a new paper to discuss on the podcast, published last week in Nature Medicine, and that's the paper we did today. It has stirred up plenty of press about gain-of-function studies and the risk-benefit, although mainly the risk of doing this kind of research, and also made some tabloid news sites that have completely overblown what the research says in exchange for a flashy headline. And she sent links to these, which was great because I used that for writing my post and mentioned a few of them today. Commentaries throughout the internet with bad headlines in it as well. She sent links to the UNC press releases, which we've talked about. So, Emily, thank you very much for doing that.
Yeah, really nice. Thanks for nothing, Emily.
No, I think it was.
No,
I mean, I might have done it at any point because it's getting a lot of press, but she kind of made me do it this week, and then I thought of having you guys on. So
her excitement level this morning for it was very high.
Great. That's great. Yeah, I told her yesterday that we were going to do this. And I do think that we need to do more of this because, we typically talk among ourselves and we have our opinions and maybe people don't care about that. But when you get the investigators in here and you let them speak about their work, I think it's more compelling in many ways.
Yeah, it's great.
But I do think the press, people need to listen to this stuff. You know, they want to ask their own questions, but they don't often ask the right ones. I also want to read one follow-up from last time. Last week we discussed 2 influenza virus papers, and one of them was from Kanta Subaro's lab at NIH, and the lead author was Seema. And I had emailed her to clarify one of the figures beforehand, and I didn't hear from her. And then I heard from her over the weekend. She said, "Dear Vincent, I apologize for the delay in responding to your question. I got your email after 1 p.m. on Friday. Wanted to make sure I had the correct answer from our collaborators, Drs. Ram Saisakaran and Akila Jayaraman, who performed the glycan binding assay shown in Figure 2b. After listening to TWiV yesterday, I realized there were some multiple concerns regarding that figure. 1, what does % binding signal mean? And 2, what is the control that we are comparing the alpha-2,3 H1N1 222G revertant virus to? First, in the glycan binding assay, Akila normalizes each of the raw binding signals by dividing it with the maximal binding signal obtained in that assay. Typically, the maximal binding signal value is obtained for the strongest binder at the highest viral titer in an assay. I believe this is as you suspected. Second, in the text, we are comparing the revertant 22D glycan binding profile shown in Figure 2b to the binding profile of the alpha-2,3 H1N1 virus that had a 222G that we had published in the 2013 paper. All right, so that's the comparison to previously published data. So it's normalized and compared to binding studies published before. I hope this answers the concerns you and your TWiV colleagues had regarding Figure 2b. Please let me know if there are any further questions. Kanta and I are thrilled that you picked our paper to discuss on TWiV, and I was intrigued by your connection to the mammary gland transmission paper that came out recently. So, Rich and Kathy, does that make sense?
Yep. That's all good.
Mm-hmm.
This episode of TWiV is also sponsored by the 32nd Clinical Virology Symposium of the American Society for Microbiology. This is a meeting taking place in Daytona Beach, Florida in May of next year, May of 2016, specifically May 19th through the 22nd. It's the 32nd Clinical Virology Symposium, and they're now accepting abstracts. Send in your abstract before March 17 to debut your latest research on rapid viral diagnosis, the clinical course of viral infections, and preventative and therapeutic modalities for virus infections, all before over 1,000 researchers and primary care physicians. Interesting audience there. For more information, go to asm.org/CVS2016. That's asm.org/CVS2016. We thank the American Society for Microbiology for their support of TWiV. And is it okay just to go to some picks now? So because we're already at an hour and 20.
Yeah.
And then it will be
We might have to do an all-email episode someday, but...
Yeah, we should do one someday, right?
Yeah.
All right, let's do some picks of the week. Kathy, what do you have for us?
I picked a link to the California State Department of Health because I went to a fabulous lecture yesterday by the director of that State Department of Public Health, I should say, Karen Smith, and it was the Susan B. Meister Lecture on Child Health Policy. She gave a really good talk about the history of public health briefly, the history of vaccination briefly, and then she talked about measles and Disneyland, legislation in California, how outbreaks of pertussis led to some better legislation with respect to vaccination for children going to school, and how timing is really important for that, and the Disneyland measles episode contributed to a complete elimination of the personal belief exemption in the state of California. And she did mention in the questions afterward that someone asked, was there any is anybody appealing that decision? And, the proposition mode of government in California where you can get signatures and get something on the ballot. She said they didn't even get anywhere close to enough signatures to get that on the ballot. So that's really good. Anyway, she talked about how press releases are static and Twitter and Facebook are active. And so the California State Department of Public Health was very active on Twitter and Facebook within 2 days of knowing that there was this association of measles in Disneyland. They had it on Twitter. And interestingly, Disneyland at first resisted this, who, us? And then they embraced it. And she showed 2 slides of Mickey quotes. They had it on their electronic boards that were really great. And I don't think you'll find them anywhere, so I want to read them to you. Thousands of vaccinated people did not get measles here. And then there was one with Mickey Mouse. And he's in one of those big protective bubbles that he might be in one of the Disneyland parades or something like that. And Mickey is saying, we can't all stay in protective bubbles. That's why Mickey vaccinates. So from going to who, us, Disneyland fully embraced the, you need to be vaccinated and come here. There's 24 million visitors a year, and a large part of them come from out of the country and places where measles is still an endemic problem. So that was really good. The web page has a really cool link on the far right to data, and you can click down and look at all kinds of data for the state about infectious disease, West Nile virus, various other things. And so that was my way to have something physical that you could look to. There's a bio of Karen Smith on the website, and just it was amazing. If anybody ever has a chance to hear her speak, don't miss it.
It's good, huh?
Mm-hmm.
Excellent.
Nice. Great. That's a really good story, too. Rich, what do you have?
First of all, since we didn't do the weather coming into this, I have to say that in Austin, Texas, it is clear blue skies, and I'm getting it. It must be it's 69 degrees, so it's a beautiful day. I have a pick from NASA TV. It's a couple of months old, but I've had it on my pick list for a while, called Meals Ready to Eat. Expedition 44 crew on the space station members. Expedition 44 crew members sample leafy greens grown on the space station. And this is about growing food, plants in space. It's a little deceptive because you see this picture up on the top, and it looks like they've got a whole greenhouse on the space station, but that's a greenhouse somewhere else. And they're growing these things. If you get into the article, you can see the conditions under which they're growing these things. They have what they call plant pillows, which I guess are bags of dirt and nutrients and seeds and stuff that they grow lettuce and other things in, including zinnias. They grow flowers, and they've been noshing on these things recently. So this is important for the future long-range missions and stuff. And so I just thought it was interesting. Sorry, Dixon's not here. I thought he might be interested as well. But I thought particularly interesting that they thought that the thinking is that growing plants in space has also, Vincent, pay attention, psychological benefits. Okay? If you're going to be up in space and going to Mars and stuff, you're going to be better off if you're growing some plants and house plants and growing flowers and stuff like that. It's good for you. So there you go.
Well, that's great. I'm not going to Mars, so I'm not growing plants on Mars, and I hope the people who do go there grow plants. Yeah. Yeah, that's pretty cool. The weather here is really nice. It's beautiful today. It's a cloudless blue sky, 14 degrees Celsius, and no rain in the forecast. Beautiful. It's actually been warmer than usual. Vineet, what's it like down there in Chapel Hill?
It is also beautiful here. It's sunny, about 60 degrees. I didn't put a coat on my kid today, and I think it was okay.
Nice. How about Kathy out in Ann Arbor? What's this?
Sunny, not a cloud in the sky that I can see, and it's 45 Fahrenheit, which is 7 Celsius.
Nice. Well, all over the place. It's pretty nice.
We're supposed to get snow tomorrow, though.
You are, really?
Yeah.
We have good weather tomorrow. This morning, by the way, guys, a stump grinder appeared on my property. So my town took down a big tree at the edge of our property. You know, there's this strip of land which is eminent domain, right? The town owns that in theory.
Uh-huh.
And this tree was a big old oak, about 3 foot in diameter, and it was dying. So they took it down. And then today the guy came with a grinder, and I was going to go out and take a picture for everyone. You know, it's this big wheel with spikes on it, and this was about an 8-inch high, 3.5 foot wide stump. He was done in 5 minutes.
Yeah,
Wow.
those are great machines, aren't they?
Unbelievable. And now he left this big pile of chips, which I have to move away this weekend. But I thought of all of you guys on TWiV because we had a stump grinding episode, right?
Yeah, we had a stump grinding episode, and that was coincidentally that revolved around Buda, as I recall. And I had lunch in Buda yesterday with Neva.
Cool.
So,
how's Neva doing? Well?
Neva's doing fine.
Great. That's just great.
And Buta's a fine place to be.
I have 2 picks for you today. The first one has to do with computer keyboards. Now, I figure I spend most of my day in front of a computer in some way, and I'd like to have a nice keyboard. And a few years ago, I learned all about nice mechanical keys. And there's an article at Lifehacker called How to Choose the Best Mechanical Keyboard and Why You'd Want to. I like a clicky keyboard that gives you a lot of tactile feedback, and I love typing on that. I have one at home. I don't have one here because it makes too much noise during a recording of podcasts. But the original IBM keyboard years ago was clicky, and one of the Apple ones was clicky too. And now they make them again. And this article will tell you about all the switches. So every key, of course, has a switch under it. And the clicky ones are called cherry switches. And they have cherry black, red, brown, blue, green, and clear. And they all have different tactile feedbacks. And some of them are good for typing. Some of them are good for gaming if you need to have a really fast pressing of the key, right? I love this stuff. So very few people will care about this pick. But there's going to be some of you out there who are into keyboards. So the one I have home is made by DOS. It's called the DOS keyboard, and it's very clicky. And I feel like I'm working when I hear that click, 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 click. So that's a cool article.
They had typewriters in the olden days.
And they clicked a lot, yeah.
And had the little bell.
Yeah, the bell, the return. I used to use those for sure. Yeah. And you'd hit the metal thing to push the carriage over, right?
Right.
That was called a carriage return. And that stuck. I have a key here.
It says return.
And if you pushed too many keys at once, it jammed all the little hammers up. Boy, there's going to be a lot of people who don't know anything about this. We need to include a picture of a typewriter as the episode image or something like that.
Well, supposedly the QWERTY keyboard was made to slow down typists so that things wouldn't jam, right? Because it's not the optimum layout of keys. And now they've stuck, of course. Although you can buy a keyboard that's arranged in the optimum way, but you'd have to learn how to use it, right? One of the best things I ever did, I think in my freshman year of high school, I took a summer typing course. And I learned how to touch type, which to this day has stuck with me. And one of the best things I ever did. Anyway, that's a cool article. And the other one, earlier this week, or last week, I was contacted by a fellow named Mike Carruthers. And Mike is out in L.A. He's been a radio guy for 20 years. He's got a radio show. And he decided not too long ago to make something called Perfect Your Podcast. It's a website, and he does podcasts called Perfect Your Podcast. If you don't know it, podcasting is really hot right now. Everybody is getting into it. And at the same time, people want to teach you how to do your podcast. So there are dozens of sites all over about how to make a podcast, how to do this and that and the other, which I find really amusing because when I started TWiV, 2008, there's none of this around. I had to figure it all out myself, which is actually part of the fun. So anyway, Mike interviewed me. He interviews people about their podcasts. And that's my other pick, episode 12, discussion with Vincent Racaniello of This Week in Virology. And he was amazed that I have these niche podcasts that do so well.
Well,
you should listen to it because he says, How do you make it not boring? And he actually had listened to an episode, and he said, it's really entertaining and easy to listen to. And he said, you have great co-hosts, too. That's what makes it really interesting. He writes here, if you think virology sounds too niche or academic, he has also a podcast on parasites and one on microbiology. Those are topics would be very easy to make boring or appeal only to academic types, but those people do not make up the majority of Vincent's audience. So if you're interested in, I talk about the art and science of podcasting with Mike. And I also told him about the TWiV bump, and he said, I really would like to feel the TWiV bump. And I said, okay, I will pick your website in the podcast. So folks, go and listen to it and show him what this TWiV army can do. We have 2 listener picks. One is from Jesse. Dear TWiV, I'd like to submit a listener pick of the week, a book called Networking for Nerds by Elena Levine. It's a great resource for academics and also those pursuing other career paths, and discusses what networking is, connecting with others for mutual exchange of value, promoting your own brand, how to interact with others and find people to interact with, and more. For those at institutions with subscriptions to Wiley Publishing, it may be downloadable as a PDF for free. It gives a link to that. For others, it's available from Amazon. Keep up the great podcasting. So Jesse is Jesse Knorr of Bacteriophiles. That's a very nice podcast about bacteria and sometimes viruses. Jesse is a PhD student down south, and this Bacteriophiles is on microbeworld.org. It's supported by ASM, so you should check it out. Give Jesse the TWiV bump.
It's spelled Bacterioffiles.
Right. That's
Not
very good, not
PHI.
PH.
Yes.
So
Yeah.
that's a nice, I really like that title. I think that's very good, as opposed to autoimmunity, which I didn't even say right.
That's right.
Audio immunity.
Yeah.
There you go, Rich.
All right, the other pick is from Russell. If you haven't done World Mapper, you should. Great series of maps, including those showing the global importance of a large number of diseases, including infectious diseases, using a unique map format. The higher the prevalence in a country, the bigger it is on the map. Really interesting to look through them, look at disease and cause of death. Russell is a professor of clinical pediatrics at Tulane University School of Medicine. And finally, last week I mentioned a contest to win a brand new 2-volume copy of Principles of Virology, the 4th edition, just published. And hardly anyone responded, which tells me nobody listens to the end.
We got some.
Couple.
I got
I
some
got a
Twitter
few
action.
hits.
Definitely.
We got a couple, right?
I would say, well, I probably had 10 people
Really?
add me. Yeah.
Yeah, at least. Problem is, Vincent, you have a really high background of Twitter followers. You gotta go with guys like me who have hardly any followers, and then I can see the signal.
Right.
Right.
Well, I just think that I can't imagine you wouldn't want to have a free, textbook set. I have a lot to give away, but I'm not going to do it on Twitter. It doesn't, it's not. I want to try to promote TWiV on Twitter, but obviously it's not working. But I'm going to let it go a little more and see what we collect, and then we'll figure out another contest to give away a book set. So what they say, of all the listeners, maybe less than 1% ever respond to anything. But I figured if you give things away, they would. You remember the Drobo contest we did? That was pretty popular.
Yeah, that was fun.
All right, you can find this and all the other episodes of TWiV at twiv.tv, also on iTunes. And if you have questions or comments, send them to twiv at twiv.tv. Today we had 2 very special guests, Ralph Barrick from the University of North Carolina, who has left us a long time ago because he has important things to do. We thank him for joining us. And a postdoc in his laboratory, Vineet Menachery, right?
Yep.
Thank you.
Menachery.
Menachery.
I hear you can call me Vineet.
Vineet Menachery. Thank you so much, Vineet.
Yeah, it was great. Thanks for the opportunity.
It was really good, and I'm sure we will hear from you again. Are you going to go to ASV next year?
That is the plan in Virginia Tech.
Yeah.
Mm-hmm.
Yep, that's the plan at the moment.
Seek us out and say hello. We'll do a TWiV there, but seek us out at some point and say hello. I'd like to meet you.
And we know you have important things to do too.
Yes.
But we're happy that you were able to stay a little longer.
Yeah.
I'm excited to be described as special. So
you are a special guest for sure. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is usually in Florida. He is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. It was always a great time.
You had a good signal there from Austin.
Oh, yeah. This is a good podcasting, podcasting place. And it turned out that the change in time was just right. This fits great into my day.
Are you at your daughter's home?
I am at my daughter's home, sitting at the breakfast table, yes.
It's really quiet there.
That's because all the kids are in school and all the parents are in work, and it's just me and my wife here.
Gosh, it is Friday.
Hanging out.
It's right. It is Friday. So you're hanging out there till Thanksgiving?
Hanging out there through Thanksgiving. Part of what we're doing is grandparenting here because the parents are extraordinarily busy. The Harper, age 7, is got a singing part in their Texas State's production of Evita, which is outstanding. I can't believe what an amazing theater program they have here. And so my daughter lit the show, and Harper's in it. And my daughter's working on another show every night, so Ibby and I are helping out taking care of the kids because it's pretty crazy around here. I've seen Evita 3 times this week so far, and I'm going to see it another 4.
Nice. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.